I'd like to thank you, both speakers, Dr. Lieberman and Dr. Suzuki, for your fascinating talks. I'll open up the Q&A session at this point. Feel free to continue depositing questions in the Q&A window. You speakers, like the other and like in the other session, I'd like to invite you, if you have questions to one another, I would like to make comments on each other's presentations.
I can start by
okay. I have a lot of questions too, but yeah, you can start. Yeah. No.
So I unmuted first, so I'm gonna go first. So so you just mentioned some horizontal strain sharing between different people not in the same family, and I was just wondering if you could talk a little bit more about what species those are or what taxa those are, because I find that surprising.
Yeah, so you're talking about the strain sharing events, say within the same town between
yeah, like within Gabon, the Gabon
yeah.
and Vietnam data. Yeah.
Yeah, so that's so actually I don't have the list, so I don't have the taxa in my head. I should ask Liam and Hagai. But for the strain sharing, yeah, so it's not on the list. Yeah, so all the list in the previous slide was showing the one that was enriched in a way in the related pair. So I don't have the list, but yeah, I'll let you know. And if that matches with the co-phylogeny list, that'll be interesting. Yeah, good point. Good point. Yeah. So maybe I'll ask one question. It was a fascinating talk, and I really enjoyed. And I know nothing about the pores or skin microbiome, but I was surprised, I guess, about not seeing any evidence of adaptation or this this priority effect. Yeah, so you mentioned pore as island, but like, do different pores, say like island biogeography, like do different pores have different size, different secretion, right, different oil? Then you can imagine it can be different, right? So I was pretty surprised.
Yeah, so, I have to admit that I don't know that much about pore physiology myself, right? And I've been trying to learn more, but there's just not a lot of data about human skin, and you can't learn that much from animal skin, right? These are weird sebaceous follicles that we're mostly studying. They're blackheads that we've sampled for the most part. So that's a big caveat. Right, and so they're like hair follicles that have been like screwed up because they have a tiny hair follicle and lots of sebum production. So we don't know much about them. It's very likely that they each are really different. One interesting anecdote that supports your idea that like pores are really different is that we're not able to get good molecular clock data for C. acnes. We can get nice molecular clock data for Bacteroides fragilis, even on 2-year time series, but I have a 5-year time course for myself that does not have any signature of time. I mean, like there's a weak signature of time, but it's like very, very weak. So I think that there's a lot of stochastic variation into like, are pores growing or not? But yeah.
In this sense, like in this respect, I wanted to ask if you saw any effect of age, making the skin more porous, less elastic.
Yeah, so we just studied a number of healthy adults around the same age, and we weren't powered to really look for age. We're looking at something very specific and weird. That's, I think we need a lot of people because there's just so many variables in what we're looking at. But we are starting now to look at least a community composition at different ages, and strain variation at different ages. We have cohorts who are children who are pre-adolescent, who have just went through adolescence, and their family members. And the data is still being analyzed, but I'll say that what we can say so far is that we don't see strains that are specific to people of different ages. Although we do, and it seems that there might be more colonizations during puberty, some more new colonizations, the environment during puberty when our strains are really changing. Sorry, when our skin is really changing, right? What happens during puberty? We have more hormones, we have more oil secretion, and that allows more colonization. Or we think that that raising of the carrying capacity might bring in more new strains, but that data is still being finished.
So does any of you have any questions to each other? Any more questions?
Yeah, I can continue, but should we answer the questions from the audience too, or?
Oh, we have time. That's okay.
Okay. Let's see. That was the major one, but okay. Yeah, so the colony-based sequencing, I agree that it's a great method, right? To basically phase these strains, like you saying. It's been a theme of this symposium, and I think it's very important. Do you think we can overcome this by long-read sequencing is one question, but also, there are some downside of colony sequencing. Say there's evolution in the plates too, right? What do you think about long-read sequencing and these caveats of colony sequencing?
Yeah, thank you so much for that excellent question. It's really important. So I'll first talk about long-read sequencing and then talk about colonies. So I'm not confident about long-read sequencing being, I think it could be useful in many cases, but it depends on the questions you want to ask. So if you just want to look for genes under selection, you may not even need long, you can do it probably from short read sequencing, and we do that in our 2014 Nature Genetics paper, where we just look for genes that are mutated, genes that have multiple alleles that are at intermediate frequency and don't seem to be happening in the same exact cell because they're not at the same exact frequency. But if you really want to build trees, there's a real problem because you can have a cell coexisting with its direct descendant, right? There's one mutation away, right? And so if you have long read sequences, unless you could maybe infer that some cells have it and some cells don't, but if they're on opposite ends of the genome, you're not really going to be able to figure that out, right? If by just using some marker that's in the middle. So you have to get lucky enough. I think long read sequencing is only going to save us if we can sequence the entire genome. And maybe that'll happen, right? Maybe that'll happen and I'll be wrong. Okay, so now why is colony-based sequencing the right way to go about getting around that, right? We'd like to do single cell, right? That'd be better, but single cell sequencing is not there yet and is introducing a lot of errors. Colony-based sequencing, you do get mutations in the lab. There's a couple ways of avoiding them. One is by just sequencing the colony right away. So don't even passage it. As soon as the colony is visible, just scrape that off and sequence the whole genome. You have to then make sure that you're not sequencing two cells stuck together and do some careful QC to make sure you're avoiding those. We do a lot of that. It takes a lot of time. But by doing that, you're only going to get a false SNP if it occurred like in the first cell division on that plate and also was adaptive. Okay, but even if you do that, even if you do some passaging and allow some mutations, you just then need to be careful about focusing on the mutations that are not at the tips of the tree, right? Things that are happening in the lab are going to happen at the tips of the tree. And so they're not going to be, you're probably not going to focus on them anyway.
Okay.
Thank you.
Thank you for the good question. I'm sorry if that was a long answer.
Okay, I think we can now move to a bit of a question from the audience. So the first question, I'm going to go by upvoting. So the first question was from our own Sean Gibbons, and he's asking, he says, really cool biogeographic patterns in these pores. Have you been able to look at the same pore longitudinally? And I think you've already answered that. And do you think these genotypes dominate in a pore for a really long time, or does this turn over a lot?
That's a great question, and I haven't answered that question. We have not been able to sample pores longitudinally. I would love to be able to do that, but I haven't found a great method to do that. And if people without really disturbing the community, but if people have suggestions, and also tracking it, right? Making sure you're sampling the same exact pore sometime later can be difficult without like doing a face tattoo. But if people have ideas, I'd love to hear them that are not face tattoos. And then I think I also will say one thing about the second part, which is, are they colonized for a long time? And we think that pores are not necessarily colonized for a long time, and that they're like, there's this bottleneck and that they're destroyed and recolonized. And the reason we think that is because we find very closely related pores, right? If pores were colonized once and then sort of dominating that pore forever, we might expect a tree that kind of looks more comb-like, where there's some sort of accumulation of mutations within that pore. since it was first colonized. But what we really see is very short recent common ancestors and a lot of sharing between pores.
Thank you. Dr. Suzuki, another question from Sean that I also found interesting. So the genome size observation is very interesting, but there was one outlier there, Aobacterium. He had this variant as this strain has a positive association for genome size versus co-phylogeny index. And do you have any ideas why this genus was an outlier? You're muted, I believe.
Sorry, yeah. So I think you're talking about the Eubacterium that's showing the
Yeah.
opposite pattern among the 8. And the question was, I'm sorry, I just blanked out. So the question
If you
was,
had any ideas why
yeah,
this
why?
genus
Yeah,
is-
so no, simple answer is no. And we really want to look at the accessory genes and, like what's special about these co-diversified strains, but we actually don't have. But oh yeah, then the one explanation for Eubacterium is it's just a terrible taxonomy that Eubacterium genus includes many divergent lineages. It's not monophyletic. So if we classify the name Eubacterium better, maybe we'll see this. And this was something we discussed, and we haven't fully resolved. It's very difficult to assign names if it's not published. So this might be the reason. So there's still a chance if we dissect the Eubacterium name, it's possible. Yeah.
Makes a lot of sense. Tami, that's the question for you. So why not do de novo sequencing of each colony rather than mapping reads to a reference, which might not be representative?
Thank you, Varun, for the question, because it's letting me get on my soapbox. And my soapbox is that when you do de novo assembly for each colony on its own, for any bacterial genome, and then try to compare to de novo assemblies to find mutations, you're going to get some mutations that are real and some mutations that are false positives, and just like you would from read mapping. But when you're just comparing two de novo assemblies to each other, you lose all the statistics of what are the reads that contributed to each allele, that which have some, right, in the statistics of 20 reads A, 1 read T, 20 reads T, 20 reads A, right, give you some idea of whether or not it's a false positive variant or a real variant. And so by using the raw reads, we get more data. And so but the second part is a very good point, which is, you got to make sure your reference genome is representative. And what we do is as I sort of mentioned, but maybe too fast, is we build once we get a bunch of colonies that are closely related, based on some clustering algorithm or another, we then build a de novo assembly from all of those. So we really can capture the whole flexible gene content. And we're mapping to something that's very closely related within 100 SNPs. So it helps us get very clean data that we can work with.
Thank you. So Dr. Suzuki, a question to you from Christian Diener. What do you think are the selecting forces for co-diversification? Are those endogenous factor like host filtering, or exogenous one like environmental factors other than diet?
Mm-hmm. No, I think it's a very open question, and there are a couple of studies that particular P. copri and Eubacterium species that people have identified population-specific strains, and they look at their genes. So these are somewhere based on isolates, and they see some metabolism difference for carbohydrate metabolism. So there may be adaptation to the local. The question said besides diet, but so there's some evidence that some co-diversified strains can be adapted to their local condition. But I think the question was more asking what's special about them compared to, I guess, non-co-diversified taxa. And I think it's very open. There are a lot of hypotheses I can think of, but I cannot support really till we do more of a functional study we're planning to do next. Yeah.
Thank you. And Dr. Lieberman, a question from Melina. how do you choose the growth media that you grow the different strain in the lab to test for fitness growth? Could it be that the skin media favors the colonizing strain, and in the lab it doesn't?
Yeah, yes, right. Anything we do in vitro is not gonna mimic things in vivo probably that well, or probably not even that good to begin with. You know, probably barely, right? And so I'm not saying that our lab fitnesses mean in vivo fitnesses. I'm just saying that if you pick a really trivial measure from in lab, you can see that strains that are taken from the same person do really, really differently. And so I think it'd be very hard to understand how that would happen, where like suddenly something on the skin would somehow, you know, they'd be equally competitive in vivo. I could be wrong, right? Maybe there's like some oxytrophies in our, or there's some missing things in our media that are really, really abundant on the skin, and there's no difference. They don't care about those at all on the skin. But I just, the differences are really stark, like, you know, 50% growth rate differences.
Thank you. Dr. Suzuki, that's a question from Hamza. She's asking, what makes GYR-B a suitable biomarker for studying co-diversification in between hominids and their respective microbiota?
I guess this is talking about Andrew Muller's work, and I think it is suitable because you're tracking the, it's one gene, but it is tracking the evolutionary history. And I guess in general, it is better, right, to have multiple markers because each gene has its own history, but I think it is a valid method. And there's a recent paper by Ackman, Nishida and Ackman, I think a month ago, that followed up that some of these captive apes obscure the pattern of co-phylogeny. So this is maybe not directly related to this question, but just a follow-up of the paper that was mentioned. They showed that maybe this geographic separation is necessary to maintain this co-phylogeny, is the most recent paper from their group. But I think as a method, I think it's a valid method for sure, if I understood correctly the question.
Okay. Doctor, yeah, do you?
I see. I see a question here that I'd like to answer.
Go ahead.
Okay. Yeah. So Jesse's asking if Jesse's saying, I'll read out loud so everyone can know, that he's seen a lot of within host evolution studies that show loss of functions mutations being selected. This makes a lot of sense, given that it's easy to break something quickly to gain a new function. But I expect these to be evolutionary dead ends, and our results are consistent with this. And do I agree, and do I care to comment? And yes, I care to comment. So yes, loss of function mutations are common all the time, and in genes often that seem to be like really critical, right? So in capsule, in Staph aureus, but also some capsule genes in Bacteroides fragilis we saw as well, and some of these like really critical sus-C importers. So I think so. The idea, a lot of people have this idea that they're dead ends, meaning that maybe that genotype is not going to now transmit to a new person. And I think that that's very possible, but I think there's another possibility that population geneticists, I think, underestimate because of how we're trained, which is very fair, which is reversion. These bacteria have population sizes that are often so large that they're sampling every possible nucleotide mutation every single generation. Right? If you're 0.1% of the gut microbiome, you're 10 to the 9th bacteria. So you're sampling every mutation every generation, and so the possibility of reverting that mutation when the selective pressure changes is very possible. And I think that it's a really interesting thing that we're interested in the lab.
Thank you. So Dr. Suzuki, an interesting question from Sean. So it's interesting that Firmicutes data at taxa shows a stronger co-phylogeny signal than Bacteroides. With so many spore formers within Firmicutes, I might have predicted that the opposite would be true. Did you look at spore forming genes where this related to co-phylogeny?
So no, we haven't looked at the spore forming present absence, and we actually didn't have any predictions at the phyla level. This is like the early days in obesity literature of a Firmicutes Bacteroidetes ratio that was associated with obesity, but we don't really know the actual mechanism, right? This is like a gross pattern, and it's very diverse within each phyla. So I think that's a great suggestion that we should look into more spore forming. Those kind of traits or genes is the next step for sure. And yeah, we haven't looked at it yet.
Great. Okay. Tami, I'm going to the next question from Christian. You're probably seeing already, I'll read it out for everyone else. Is the fitness growth rate correlated with the prevalence of a particular strain? Feeder strain occupy more pores as an individual, or are there other mechanisms, better at invading pores, better dispersal, that dictate the occupancy prevalence of different pores?
Yep. Actually, I don't think we did a formal analysis of that question, which is a good one, which is the abundance of the strain on the person, basically, and its fitness in the lab. I'm not sure we haven't done that. I think it doesn't strike. I think like it's kind of, you look at the data and the most fit strain is not the most abundant strain. But I think we should do a more formal analysis. So thank you for prompting that. And then, yeah, I think there is a sort of natural idea that if you're better in if we think that invading a pore is really what's going to determine your population size, then yeah, there should be some selection at the level of invasion. And that's a really great question. We don't see much evidence. Oh, sorry. We can't get evidence of it right now. We don't have a very good system to mimic invading a pore. Wait, there was another idea. One thing I want to mention that I don't think I drove home is the flexible gene content of these bacteria is not that different. So Staph aureus, they have very different flexible genome from one strain to another. And a lot of the bacteria we've been discussing today, including Bacteroides fragilis. But C. acnes is kind of a weird bug in that it's got a really core genome. There's a relatively large core genome. There's maybe 10% strain variation from one to another at max. 10% gene content variation. And so, when we see things are more fit or less fit in the lab, it's unclear if if that's adaptive or it's just like sort of some neutral loss of function mutations. I have this idea in my head that I think is maybe a little extreme, but I want to just put it out there as an idea, which is that C. acnes is evolving almost completely neutrally at this point. That the strains are almost exactly the same, and that in the lab we can see differences, but they're not super relevant. And yeah, which I guess kind of relates to this idea that maybe some of these differences in the lab are alleviated. I want to it reminds me of one other point I didn't make. And so I'm sorry if this is a little tangential, which is that if these pores may not just be limiting competition, right? But they may be also limiting evolution as well. And if they're limiting evolution, there's this idea that maybe pores have been designed by our hosts, by us, not designed, evolved over time, selected on over time, to select on not letting the microbes compete that much and evolve. Okay, that's just an idea. Just want to put it out there that maybe these pores are designed to limit bacterial competition and evolution. I'm not sure that I think I went off on a rails there. I'm not sure, but I think it's a very interesting question. And I think if anyone has ideas about how to better model skin invasion of pores would be great. Yeah.
Okay. Dr. Suzuki, would you like to respond to that?
No, I'm sorry. I was reading a question just came up that looks very interesting. If I can pick one.
Sure. You're referring to Bruce Mitchell?
Yeah, this one from Hamaza Amar.
Oh, okay, yeah.
I'm gonna answer. Oh, I'm sorry. I misread the name. So Mohammed Saran. Sorry.
Okay.
Well, I'll answer this first.
No problem.
So asking, where do you think the ancient European microbial genomes, like 1,000-year-old, will fit in the phylogeny? Like the base of the whole tree. So where does it fit these? I think this means like ancient. So if we can go back in time and sample a genome is how I took it. And we actually did this. So I didn't talk about it. I have a slide that I can show, but Alex Kostick's lab from Harvard published this paper on paleofeces from Native Americans. And so these like 1,000-year-old Native American feces mags were recovered, and they directly gave us the mags for at least 5 species that's high quality that we can fit in the tree. And then 4 out of the 5 clustered right next to the East Asian strains and closer to the Native American strain. So it does reflect the migration history from Asia to the Americas. So I guess for this specific question, I think it should look the same. I think Europeans should cluster with the basal of the European clade is my guess, because African strains will be more basal is my guess. So it's kind of indirectly answering the parts that I couldn't present. Yeah, but thanks for the question. Yeah.
Thank you. Tami, Priyanka from ISB is asking, does the host immunity play a role in adaptive evolution observed in your study?
Yeah, so we see these strong adaptive sweeps in Staph aureus, and these sweeps could very much be driven by the host immune system. And in fact, that was one of the hypotheses we had starting, which is, Staph aureus normally lives in the nose where it's not getting that much inflammation, and now it's going to be living on these. Then presumably it's gone from somebody's nose, probably to a kid's lesion of AD. And so there's more inflammatory environment, more higher, immune environment, and how are they going to adapt? And so very possible that they're losing these capsules because the immune system is attacking capsule. We know that is true, that we can make antibodies to capsules. We tend to think antibodies attack proteins, but they can also attack sugars, including capsule sugars. So yeah, so it's very possible that there's host selection just to remove these, and that's most of what's going on. Right? We don't know. Yeah.
Thank you. Dr. Suzuki, I'm going to Siglander's questions. So can you tell us more about Prevotella capri? It doesn't seem to fit your model, and it does seem to have something with that to do with diet.
Mm-hmm. So I think this part of it doesn't seem to fit your model part. I think I'm talking about the tree that was pretty complex, but I think it does fit if there's a multiple transfer events from Africa to others. So there's different waves of migration, and there's a paper actually that the Prevotella capri already diversified in Africa before out of Africa migration. So that's why it looks more complicated, that there's multiple transfer events. I think it's consistent. And one thing I found interesting for the strain sharing results that Prevotella is dominating the list, I think all the mother-child strain sharing papers so far, I think, are all done in Western populations, I believe. And Prevotella capri is famous for being not much abundant in Western population, and they're abundant in Africa and even in Vietnam more than Europeans. So that's why it was surprising that you see these strain sharing events from mother and child. The strongest evidence was Prevotella, if we have African samples, right? African mom-child pair. So that was a interesting finding that matched the co-diversification project, and that's how we merged this project, because it was a nice story. So we think this is one of the strongest species that we find in the list. So if you can expand more on the it didn't fit your model part, I'll be happy to answer more. Yeah, thank you.
Thank you. Tami, this is a question for you. I'm looking at the first one over there, unless you want to answer a different one. Do S. aureus and S. acnes strains coexist in skin pores? Is the S. aureus strain same in different pores, but S. acnes is different?
Yeah, so I'm sorry if that wasn't clear. C. acnes we think is really the dominating pores, and we think that there's not that much else within pores. When we sample from pores and do 16S sequencing, we sometimes get other bacteria, not just C. acnes. But I can never tell if when I'm sampling a pore, if I'm just getting the surface or if I'm also getting deeper. in the pore, or sorry, if I'm also getting, if I'm just getting what's in the pore, if I'm also getting a little bit of surface contamination. And based on like what I've seen from some really coarse imaging studies, I feel like most of what's in the pore is C. acnes. But it's very possible that other stuff is in there too, but it's not usually Staph aureus, right? The other things you get are Staph epidermidis, maybe some Corynebacterium. Staph aureus is not really a skin commensal. When it's living on the skin, it's living on the skin of people who have disease like AD. And in that case, it's not really on the pores. It's like kind of, I don't know where it is, but I think it's just kind of everywhere. And you know, in between the cracks of the cells, and then starts forming lesions, and it's really on lesions. So I think it's in a completely differently structured environment.
Thank you. Dr. Suzuki, I'm looking now at Bruce Mitchell's question. If you saw it, let me read it out. So he's mentioning vertical versus horizontal. So Francis Collins has publicized horizontal transmission as bacteria selecting genes from their environment to add to their genome. As Suzuki, Dr. Suzuki clarified that horizontal here means picked up from the environment, but apparently associated with standard neutral selection, not with the Collins horizontal plucking of target genes to add to the particular bacteria's genome. Is there a Collins style horizontal selection that is found also in microbiome?
I'm a bit confused of the question. I guess, is it mixing horizontal gene transfer?
I think so, yeah.
Transferring. So if that's the case, so we didn't look at the horizontal gene transfer in this study. So yeah, I guess I couldn't understand the question. So if you can clarify the question, I think I'll be happy.
It was referring to horizontal gene transfer, which we're not looking at. So I'm
okay.
Unless Bruce, unless you can clarify your question.
Can I?
Skip it.
Oh,
yeah.
I think the sound is bad. Yeah, if you can clarify, great. And I can answer this quick one from Trong van Nguyen.
Okay.
What, how, rank cophylogeny index, which cophylogeny tests are highly significant? And this is interesting, interesting meaning. What we did is we first sorted by the cophylogeny index. When it's tied, we're sorting by the most conservative test, which was phytools using consensus trees. Conservative meaning the least number of significant p-values. And the difficult part for us was this PACO and Parafit, these two other tests gave us super significant results. And it wasn't useful because everything was significant. So that's why I didn't explain, but we collapsed strains using this cut tree function in R to try to merge the strains that are very similar to each other to increase the power. Because if you have one-to-one matching, so these tests are not made for this gigantic trees. Yeah, this was also mentioned by Akman in the recent paper. So we tried to merge the strains that's similar to try to have the power to see some gradient of p-values for the PACO and Parafit, but phytools was the conservative one. So we sorted by the order of conservative p-values. And there are different ways to sort. This is just one way, but that's what we did. Yeah.
Thank you. We're already at the end of our time. Thank you both for these fascinating talks as well as the Q&A session and discussion. So this concludes our last session of the symposium. Thank you both again, and I would like to invite Sean Gibbons now for closing remarks.